All right, we are recording. So we're going to go ahead and I'm going to walk you through the steps to look at the structures on the eye. So this is a sheep eye, and we can identify some various features of it that we're relatively familiar with from class. We see this here is the cornea. Other than that, and each eye is going to vary a little bit with the amount of, we call it just junk. It's really the extra tissues that are attached to the eye when they're removed. We're going to be cutting a large majority of this off, so the first thing that we're going to do is I'm going to show you what you cut off and how to do it, the best way to do it. And you're going to use your scissors, and you basically just grab a couple fingerfuls like that, and you're kind of pushing the eyeball away from your fingers at the same time, and you just grab your scissors and you kind of push the eye that way, and you cut more so towards your fingers. Just be careful not to cut your fingers or puncture your gloves. And you just basically continue that, and you remove this extra. It's the periorbital fat, really, and you see the dark stuff there. Those are extraocular muscles. You don't have to identify those on the cow eye, ladies, please. You don't have to identify those on the cow eye, just on the models, or sheep eye rather, sorry. So there, once we get that, the majority of that stuff trimmed away, it looks more or less like an eyeball. One point I want to caution you about, to be careful with when you're trimming, is towards the posterior aspect of the eye. If you look here, right there, and you'll feel it with your fingertip. It'll feel like a little stub, a little bump. That is the optic nerve. Try not to cut that off when you're trimming. And if you did, don't worry about it. There are plenty of eyes if you want to redo the dissection, and you can just look at it up here as well. All right, once everybody gets to this point, we're ready to kind of walk through, and we'll do the same thing that we did in the lecture. I'll walk you through the tissue covers. So we talked about the protective structures, eyelid, lacrimal apparatus. Not going to see those here. Obviously, I just cut off the eyelid here. But we can start with the conjunctiva. And the conjunctiva, as I said, is that clear tissue that covers the anterior 1/3 of the eye. And you can see that this posterior part, let's see if we can, here, this posterior part of the eye, it looks a little bit more opaque, and this is extra shiny, slimy. Okay, and if you grab your forceps and you pick this up, that's conjunctiva. So that is the conjunctiva, and technically it's the ocular conjunctiva, but that's what we see there. Okay.
That's the thicker part that's on the front half of the black eyeball?
It's not necessarily thicker. The conjunctiva itself is this thin layer of tissue that covers the sclera in the anterior part.
That's what I'm pulling on. Okay.
Yeah.
It seems like the thicker part.
Okay. So I'd really rather you just kind of, you can be walking along, going with it, but just for the sake of time and for the video, just to kind of, I'll show you everything, and then you can go back and go through it on your own. Sorry, that's what I meant by walkthrough. And there are some steps where you're going to have to do, and I'll kind of point those out to you. And the most important, I want to make sure that you do the incision part with me, or at least after I tell you how to do it, because that's the one part that can kind of increase the we'll say danger. If there's a sense of danger in dissection in this course, because if you don't do it properly, you can cause the fluid to squirt out at you. And I actually had that happen to a student. She wasn't doing it, but the person who was doing the cutting didn't do it properly, and the fluid squirted out, and it went into her eye. And she was wearing glasses. You know, they were just regular glasses, but the fluid squirted out into her eye. She was okay, but you know, she had to wash her eyes out with the eye wash back there, and she had to go to the health services. And went to her doctor, you know, and everything was fine. Just some minor irritation, but something we would like to avoid, and it's very easily avoidable by following these simple instructions that are in your lab manual, and I'm going to give you as well. So what we're going to do is we're going to make an incision that goes around the eye like that. We need to find a point to start the incision, and you're going to use your scalpel. You're not going to cut into it. You're not going to saw around the entire eyeball with the scalpel. You're just going to make a little tiny slit with the scalpel. And the best place to do that, you find it by taking the tip of the scalpel and just kind of tap it. Don't puncture through, just tap. So I'm just lightly tapping on it, and you'll feel it. You'll feel how it's a little bit more stiff or more rigid back here, and then you'll feel it kind of give way, and it's a little squishy right here. Okay? You want to go right on the border, right in the middle between the squishy and the hard. So not too far forward and not too far backward. And if anything, I'd rather have you too far backward than too far forward, because if you go too far forward, you're going to cut through the ciliary body, and it's going to become rather ambiguous for you to see. So once you find that point, and again, wait until you see me do it. Once you find that point, okay, you're going to set the tip of the scalpel on there, and you might have to reposition your hand on the scalpel a little bit to hold it more like a, you know, like a shiv or a shank, if you will. Please don't stab anybody with it. Okay. So once you find that point, you kind of reposition your hand, and then you're going to roll the eyeball so that when you make the penetration, when you penetrate through the sclera, anything that squirts out is going to squirt directly into the tray. Okay, if you hold it like this and do it, you're projecting it towards your eyes, yours and your lab mate's eyes. So find that point, roll it over, and then when you insert it, you might have to use a little bit more force than me because your blades aren't super sharp. Mine is literally brand new out of the package. So you just kind of, just like that, and you'll see some fluid squirt out. That fluid that you see squirt out there, that's the aqueous humor. So go ahead and do that. Make that slit, and you should see the fluid come out. And like I said, you don't have to bury the scalpel into the eye. Just try and stay as superficial as you can while still making that little slit. Okay, so once you get that slit, you're going to then pick up your scissors, and if you lost track of it with your eye, and you lost sight of it, okay, find it again just by, you know, again, lightly palpating. You'll see it separate a little bit. Again, don't penetrate deep or directly into the eye, because you might pop the vitreous body, and then that kind of makes the whole thing hard to see. You'll see the vitreous humor, but you won't get to really see the body. Okay, when you find that slit again, you might need to reposition your hold on the eye. You're going to cut along, around the circumference of the eye, but you're going to keep the blade of the scissors as parallel to the sclera as you can. So like this. And you just complete the cut all the way around. And some more fluid might spill out. Yeah, these scissors might be a little dull. And if you run into a part where it becomes really difficult to cut through, you might need to navigate more towards the anterior portion because you're cutting through the sclera, or maybe the scissors are just really not doing their job. And then just take small little bites with the tips of the scissors. It'll cut through. There, is that better? Okay. Okay, so I'm just going to leave mine kind of attached so I can hold it like this. And when you open it up, and I think it's something more that I've seen with these sheep eyes, the vitreous bodies, I haven't really been able to find a good one. If you have something in here and it looks like a little pillow, like a little yellowy jello pillow, that's the vitreous body. And this film, but if it's deflated like it is here, this film, this is the retina. Okay? And this right here, and I just stabbed the tweezer, that's the lens. That normally sits right here. Okay, so now that we've got the dissection part of it done, I'll go ahead and I'll walk you through the structures, and if you did, everybody should be at this point, you can kind of follow along. So I'll just kind of cap this back up, and then if you have it completely separated, it doesn't matter which half you use, front or the back. So we identified the conjunctiva, right, and the conjunctiva lies over the sclera in the anterior 1/3 of the eye. The rest of this that we see back here, it's covered by some extraocular muscles still a little bit. Didn't quite clean it well enough. Remove that. And you see there, I just tore that extraocular muscle off right there. That is sclera. So that's the hard part. If you were to poke through, the sclera is the first thing that you're going to go through in the posterior aspect. But if you come to the anterior aspect, technically the first tissue you go through is the conjunctiva. But that's not the case back here. Okay, so that is the sclera, and if you look at it from a coronal view, here, you see that, I'll zoom in. You see this thin white layer? Okay, all the way around, that's the sclera. And if you take your forceps, or if you have one of the sharp probes, you can do that as well. Forceps work if you just kind of clamp them together and carefully tweeze away a small portion of that black. You should be able to kind of pick it up and get behind it like that. There. So this right here is the choroid. So that's the vascular layer.
The black part is the choroid?
Yes.
Okay.
The black part is the choroid, and then you see, tip that over a little bit. Right there, that's sclera. So if you've ever seen a tennis ball and chopped in half, it's kind of like the inside of a tennis ball. Okay, so we'll go ahead and put that back. In this case, like I said, the lens really should be sitting right there. It's still attached to, you have the vitreous body?
Yeah.
Okay, leave it, because I want to use that as an example in a minute here. So here's the retina.
Move it up again.
Oop, thank you. So here's the retina, and it might be kind of disheveled, meaning kind of crinkled up and looks like it's bunched up in one area. You can put it back by gently manipulating it with your forceps, or maybe even better, the blunt probe. Okay. If it gets too messed up, it's going to look like a big pile of gobbledygook, and you can't do much with it. But that's what that is. That is the retina. And normally, it's a thin layer that lines the on the choroid on the inside. Good. Alright, so now I'll kind of come back up here to the anterior portion of the eye, and we'll do like what we did in lecture. We'll trace the path of, a light ray as it passes into the eye. So, ray of light enters in through the cornea, so that's this outer layer right here, and normally it is clear, but obviously this animal's long gone, and the specimen has been preserved, so that kind of incipitates some clouding of the cornea, but normally it's crystal clear. You'd be able to see right through it. And now I'm gonna do something. You don't have to do this because it tends to kind of increase the danger of injuring yourself a little bit. I'm gonna cut through the cornea so that you can see the iris. I don't want you to see. I'm going to do this. You don't have to do it. Like I said, it gets to be kind of tricky, and if you slip, and especially your tools being so dull, you'll have a hard time doing it. And if anybody wants it done on their eye, I can come around and do it for you. All right. So you see I cut a small little window, so a small portion of the cornea, cut it away. Zoom in a little more and focus. Okay, and you can see here is the iris. So I didn't really talk about what the iris did. I was kind of getting there in the lecture. The iris is another layer of smooth muscle, and it's a diaphragm muscle. Okay, that will constrict and dilate. Okay, so when it constricts, it makes the size of the pupil smaller. That's really big. So it makes the size of the pupil smaller, and when it constricts, it makes the pupil smaller. When it dilates, it makes the pupil larger. So that also goes along with visual accommodation, so kind of adjusting to the conditions of the environment or the amount of light. Because obviously if you've ever had a flashlight shined in your eyes, okay, that's a lot of light, and it overstimulates all of the visual receptors that you have, and you can't see anything. That's all you see is white light. And your pupils would obviously constrict in response to that. But this side of it here is the iris. Now the reason why I do it this way, because when we look at the other side, this whole thing, over there, so you can see it a little better, this whole thing is the ciliary body. So remember I was telling you in lecture, the iris, imagine the iris on one side, ciliary body in the other. They're kind of smooth muscle layers that are pancaked on top of each other. Okay, so iris is here, ciliary body is posterior to it, and it goes all the way around the circumference of the eye.
So is that all just one big muscle?
Like I said, the ciliary body is made up of those little individual parts, the ciliary bodies. I can't really explain it any better than that, but there are little individual units that have attached suspensory ligaments. So each of those, and it's kind of hard to see the suspensory ligaments on the cow eye. You can kind of infer where they would be. Kind of looks like the gills or the underside of a mushroom. I don't know if you've ever seen a mushroom, like the finny gill area. Okay, each one of those lines is a suspensory ligament, but it's pretty difficult to, actually near impossible to, from a gross level, to dissect a single suspensory ligament. But that's what they are. Okay, and you can see remnants of them on the lens.
So that means they insert on the lens?
They attach to the lens, correct, but just to the edge of the lens, right here. So you see where there's a little bit of, looks like pigmented tissue here on the lens. Okay, the suspensory ligaments are kind of embedded in that, and they're attached to the lens so that when the muscle of the ciliary bodies contract, okay, it causes the shape of the lens to change. So again, we'll kind of step back to our process of light entering through the into the eye. So we went from the cornea, and here, thank you. You'll see an empty space that's posterior to the cornea, and your lens would sit here. That's the anterior chamber. And if you had the vitreous body intact, which I think you did, I want to show you what that looks like. Perfect, thank you. So they still have their vitreous body intact, and it will literally look like a little, almost like a jellyfish. Zoom out a little. So you see that right there. If I can carefully get it to come off of there. Oop, there we go.
Gotta go up again.
I will, just a second. There we go.
It's like an egg.
Yep. Yeah, that's a good analogy for it. Like an egg. And the lens sits directly on top of it. Okay, it's not really attached to it, but it's not going to come off without breaking the very delicate membrane of the vitreous body. So the vitreous body is what holds the fluid in this structure. That fluid is vitreous humor, and the structure itself is the vitreous body. So without the humor, there is no body. Okay, without the body, what's the point in having the humor there? And you see the lens, and you can really see, I'll zoom in some more so you can really get a good look. There we go. So you see those little individual lines. Each one of those is a suspensory ligament attached to the lens. And yeah, their retina was a little more intact than mine. Slide that out of the way. You see there. So you can see the layers. Sclera, choroid, and then retina. A lot of times students ask about that shiny layer. Looks
The inside of an oyster.
yeah, it looks like shiny blue. I don't think it's the same as that, but very similar in appearance. That's something it's called the tapetum lucidum. It's not found in human eyes. It's only found in animal eyes. And if you've ever seen an animal in the headlights or with a flashlight, what happens to their eyes? They glow, right? Some of them different colors, green, red. The reason for that is because the light reflects off that shiny layer, and it increases the animal's night vision. Okay. Humans don't have this layer, so when light enters our eyes, what color reflects back? Think about taking a picture. Red. And what's red in our eyes? It's also found circulating throughout the rest of your body? Blood. So that's the only color that's going to be reflected back, so that's what you see. That's why you get the red eye.
Is the human eye smaller or about the same size when you pick it up?
I would have to say it's definitely closer in size to the sheep eye than the cow eye. Cow